Hello everyone and welcome to my channel. In this lecture we will be discuss the general characteristics of Rickettsia. The Rickettsia are obligate intracellular parasites. Obligate means without their host they cannot grow or they cannot multiply. Intracellular means they are replicating inside the cell of host. Obligate intracellular parasites of the arthropod vectors like plias, lice, ticks and mites. These are nothing but a gram-negative pleomorphic rods. The transmission of Rickettsia does not occur from one human to the other human. For transmission of Rickettsia, they require a vector or they require a carrier, and these vectors are the arthropods like plia, lice, ticks and the mites. The name of Rickettsial, these are the gram-negative bacteria, but we know that the one disease, that is a rickets, the causative agent for rickets is not a Rickettsia organism, but the causative agent for rickets is the deficiency of vitamin D. So don't confuse, the disease rickets is not because of a infection from a Rickettsia, it is because of infection from a deficiency of a vitamin D. As the Rickettsia are obligate intracellular parasites, in past these bacteria or these organisms were placed in between a viruses and the bacteria. There was a confusion whether these are the bacteria or whether these are the viruses. The scientist Howard Taylor Ricketts, this scientist studied a disease, that is a rocky mountain spotted fever in a Bitrot Valley of Montana. In honor of this scientist Howard Taylor Ricketts, the name for a genus is given as a Rickettsia. The scientist Ricketts, he just injected the culture of the organism himself with a lot of time, and he got injected more times. The scientist Ricketts became interested in a strain of typhus known as a Tobardillo, and because of this injection of the organism into his body, he himself died of this disease. General characteristics of the Rickettsia. Rickettsia are similar to a gram-negative bacteria. They contain DNA and RNA as their genetic material. They show a presence of enzymes which are required for a Krebs cycle or a citric acid cycle. They show a presence of 70S type of ribosome for a protein synthesis. The antibiotics like tetracycline and chloramphenicol, these two antibiotics are antibacterial antibiotic, and these antibiotic or such antibiotic can inhibit the growth of Rickettsia. So because of these some characteristics, presence of 70S ribosomes, enzyme for a Krebs cycle, presence of DNA and RNA, and a gram-negative morphology, and inhibition because of antibacterial antibiotic, the Rickettsia are classified under a bacteria. Before that, they were thought as they are viruses. Some such characteristics are there why these are taken as viruses. because of their small size, less affinity towards a gram staining, grows only in a cytoplasm of eukaryotic cell, and as these are obligate intracellular parasites. So because of this small size, less affinity towards a staining, growth in a or multiplication in the cytoplasm of host eukaryotic cell, primarily these organisms were classified under a viruses, but actually these are a bacteria. The survival of a Rickettsia depends on entry, growth and replication within a cytoplasm of eukaryotic host cell. That's why they cannot live in an artificial nutrient environment and is grown either in a tissue or a embryo culture. For a growth, we know that for a growth of viruses, there is a need of a live cells. But this is a contrasting characteristics of a Rickettsia. Rickettsia does not require any live artificial cells. For cultivation, we can make a use of a tissue or a embryo culture. The vectors or reservoirs includes animals and arthropod vectors. Humans are the accidental host. The cell wall of Rickettsia is composed of peptidoglycan and a lipopolysaccharide, and this characteristics is similar to a gram-negative bacteria. Presence of lipopolysaccharide and a peptidoglycan. This is a characteristics of a gram-negative bacteria. The Rickettsial consists of three genera: Rickettsia, Ehrlichia, and Coxiella. Three genera are there: Rickettsia, Ehrlichia, and Coxiella. On the basis of intracellular location in their host, they are of three groups. One is the typhus group, spotted fever group, and Coxiella and Ehrlichia group. The typhus group, they are located in a host cell cytoplasm. A spotted fever group, they are located in a nucleus of a host cell Coxiella and Ehrlichia, they are located in a cytoplasmic vacuoles of the host cell. Here are some images of the Rickettsial species. In this image, we can see for the typical cell of a Rickettsia. Part A, this shows a typical cell of a Rickettsia. Here, this is a slime layer of the Rickettsial species. In a B part, here the host cell is a chicken embryo cell, and within that host cell, the Rickettsial cells are there. This is a chicken embryo cell. This whole is a chicken embryo cell. This dotted spot or these spots are scattered Rickettsia. This is a mass of a Rickettsia in a nucleus. Here are some species, their disease and their vectors, by using which vector they get transmitted from the animal to animal. There is a need of a reservoir, or there is a need of a vector, or the need of reservoirs. A species Rickettsia prowazkii, this causes the epidemic typhus and Brill-Ginser disease. The Rickettsia prowazkii use the host, that is a lice. The Rickettsia typhi, it causes an endemic typhus. The vector for transmission of the Rickettsia typhi is a rat flea. Rickettsia rickettsii, it causes a RMSF disease, that is a Rocky Mountain Spotted Fever. The vector for transmission of Rickettsia rickettsii is ticks. Rickettsia conuri causes botonous fever. The vector for transmission is ticks. Rickettsia australensis, it causes Australian tick typhus. Vector, that is ticks. Rickettsia siberica, it causes Siberian ticks typhus. Vector, that is ticks. Rickettsia acari causes a rickettsial fox, and they uses mites for their transmission. Pathogenesis. After infection, during the first day of an incubation period, local reactions are caused like hypersensitivity, and some allergic reactions are there, and this is because of the products which are inserted by vectors like ticks or mites. After biting of arthropod vector to the host, the arthropod vectors inject the bacteria from their saliva into the circulatory system of the host, and where there is a entry of a bacteria into a circulatory system of the host. Here, at the site of a biting, bacteria multiply and they disseminate via lymphatic system. Further, the host immune system uses a phagocytosis by a macrophages, and these are the a barrier to a rickettsial multiplication. Macrophages are a defense mechanism. By a phagocytosis process, the host body start to kill the entered bacteria, where the bacteria get phagocytosed. If phagocytosis does not kill the bacteria, after 7 to 10 days, the bacteria or the organism disseminate and replicates in a nucleus or cytoplasm of host cell. The infected cells shows intracytoplasmic inclusions and intranuclear inclusions. These are the bodies, or these are the inclusions: intracytoplasmic inclusions and intranuclear inclusions. The inclusions which are present in a cytoplasm are called as the intracytoplasmic inclusions, whereas the inclusions which are present in a nucleus are called as the intranuclear inclusions. After some day, multiplication of the bacteria then leads to damage of endothelial cells and causes a vasculitis. The symptoms like development of maculopapular skin rashes, necrosis of perivascular tissue, thrombosis and ischemia. These are some symptoms related to the rickettsial infections. Disseminated endothelial lesions lead to increased capillary permeability, edema, hemorrhage and hypotensive shock. Because of disseminated endothelial lesions, there is an increase in a capillary permeability, edema, hemorrhage and hypotensive shocks. Endothelial damage can lead to activation of a clotting system. The disseminated intravascular coagulation, that is a DIC. Here in this image, here there is a infection, or there is one cell of a Rickettsia. This one is a target cell nucleus or a host cell nucleus, and this cell is called as a elementary body or a EB. This cell is a EB, This elementary body, after entry into a host cell, get increase in its size and converted into a RB, that is a reticulate body. This is a reticulate body. This reticulate body, after cell division or after a binary fusion, it get increase in its number. The number of RB or a number of reticulate body get reorganized and leads to formation of a elementary body. Here are the elementary body or a number of elementary body, and this number of elementary body lyses a host cell, and after lysis of a host cell they get released into a environment and ready for a further infection to a new host cell. This is a life cycle of the Rickettsia by a formation of elementary body and a reticulate body. Here the same life cycle is there. Here are the EB or elementary body which are released from a host cell after a lysis of host cell. From this point, a small sized EB is there. EB after increase in cell size get converted into a RB. In a RB there is a one endospore. This RB after a binary fusion forms a EB or a elementary body. Here after a 12 hours around a EB there is a formation of a inclusion membrane. The EB after a maturation or a multiplication increase in its number and lies a host cell and get released from a host cell. Now here are some infections caused by a Rickettsia. Typhus fever group causes a disease like epidemic typhus or a vril ginser typhus, endemic typhus, spotted fever group, Rocky Mountain spotted fever, cybernetic typhus, Boutonus fever, Australian tick typhus, Rickettsial pox. Epidemic typhus, or for epidemic typhus we can say a classical typhus. The causative organism for epidemic typhus is a Ricketsia prowazki. Vector for a Ricketsia prowazki are a louse or a lice. Incubation period 5 to 21 days. Incubation period means it is a time or it is a period after entry of a pathogen into a host body to showing a symptoms. For epidemic typhus, incubation period is 5 to 21 days. The mortality rate is 20 to 30% in an untreated patient. Symptoms: severe headache, chills, generalized malaria, high fever up to a 39 to 41, vomiting, macular rash after 4 to 7 days, and unconsciousness. These are some symptoms: headache, chills, generalized malaria, high fever, vomiting, macular rash, and unconsciousness. The Brill Ginser typhus. This occurs after the person who is recovered from the epidemic typhus and reactivation of Rickettsia prowazki. In this case, the organism can remain as a latent, and after some time it get reactivate and it show symptoms similar to that of epidemic typhus, including a maculopapular rashes. Here, in this case, there is a low mortality rate and a mild illness. Then the endemic typhus or a murine typhus, the causative agent for a endemic typhus is a Rickettsia type. The vector, that is a rat flea, when the infected rat flea bite to the healthy host, then the healthy host got a infection. The murine typhus is under a recognized entity and it is confused with a viral illness. Most people who are infected or who got a disease do not realize that they have been bitten by a fleas. Symptoms of the endemic typhus: headache, fever, muscle pain, joint pain, nausea, vomiting. 40 to 50% of the patient will develop a discrete rash after 6 days, whereas up to 45 patients develop neurological signs such as confusion, stupor, seizures, and imbalance. Then next, RMSF, Rocky Mountain Spotted Fever. The causative agent for Rocky Mountain Spotted Fever is Rickettsia rickettsii, and the vector for this disease is a tick bite. Incubation period is near about 1 week. It is a serious disease. It is similar with a typhus fever, but here the rashes appears early. The initial symptoms are fever, nausea, emesis, that is vomiting, severe headache, muscle pain, lack of appetite, parotitis. Later signs or the later symptoms are maculopapular rash, petechial rash, abdominal pain, joint pain, and forgetfulness. Rickettsial pox, causative agent for the Rickettsial pox is a Rickettsia acari. Vector for this Rickettsial pox is a mite. It starts with a benign febrile illness and ends with a fascicular rash, and these rashes are resembling to the chicken pox, so the name is given as a pox. This disease is non-fatal, self-remitting. The first symptom of this disease is that after biting of the mite, there is a formation of a bump, and this bump get blackened, or it shows a crusty cap-like structure. The many of the symptoms are similar to that of the flu, like fever, chills, weakness, achy muscles, and rashes. Here are some images of the macular rashes. This image is of a macular rash, and this image is of a late petechial rash. Complication of a rickettsial diseases: bronchopneumonia, congestive heart failure, multi-organ failure, deafness, disseminated intravascular coagulopathy, that is DIC, myocarditis. Myocarditis means inflammation of heart muscle, endocarditis, inflammation of heart lining, glomerulonephritis, it is a inflammation of a kidney. These are a complication of a Rickettsial diseases. laboratory diagnosis. For laboratory diagnosis, the host or a patient's different kind of specimens are collected and checked for a presence or absence of particular species from a Rickettsia. For this specimen, culture is isolated and it is performed for a number of serological tests. Culture and isolation. The blood of patient is inoculated into a guinea pig or a mice. After 4th week, if the animal like mice or a pig responds, then it can be concluded that which species is there. Symptoms in that pig will be fever, scrotal inflammation, swelling, necrosis. For a detection or for a diagnosis, there is a one important test, that is a well-Felix test. In a well-Felix test, here is a antigen and antibody reactions. If the patient is suffering from any kind of a rickettsial infection, that patient's serum will show a presence of antibody against it, and by using a particular antigen or a particular half killed pathogen, that is an antigen, we can diagnose whether a patient is infected with Rickettsia or not. Other tests are there: complement fixation, indirect fluorescence test, or a direct inflorescence test. Treatment: how to cure the patient who got the infection. At an early stage, there is a use of a number of antibiotics. After some day, if the patient is not going to recover from a treatment with an antibiotic, the drug or antibiotic like doxycycline, and this doxycycline is better than a tetracycline and the other protein synthesis inhibiting antibiotics. Chloramphenicol, it is a substitute for the tetracycline. The fluoroquinolones, this is a group of antibiotic fluoroquinolones loans such as ciprofloxacin or floxacin can be used for a treatment of the patient. Thank you very much. If you like my video, subscribe my channels and share my videos. Thank you.